Namaskar, I am Biju Dharmapalan and you are watching Science Time, a show that brings you the latest developments from the world of science and technology. Let's first look at some of the exciting stories. National Brain Research Center researchers moving closer to Alzheimer's Disease therapeutics. IIT Roorkee researchers develop biopolymer-based edible coating for horticulture products. IISER Pune Researchers develop a sophisticated microscope to understand the structure of protein titin. Now let's see the news in detail. Scientists from the National Brain Research Center, Manesar, Gurugram, led by Dr. Pravat Mandal and his team, have presented a novel working model for Alzheimer's disease. The model is based on intense ongoing clinical research since 2009 at NBRC in collaboration with Dr. Manjari Tripathi of AIIMS, New Delhi. Dr. Mandal and his team have found that brain oxidative stress enhancement at the hippocampal region is the early event and subsequent amyloid beta oligomerization along with amyloid plaque formation occurs in the brain. Briefly, in the brain of Alzheimer's disease patient, there is significant depletion of glutathione in the brain hippocampal region. Then, the brain amyloid beta peptide is exposed to radicals and sensitive residues. The methionine 35 is affected by these radicals and initiates the structural modification of amyloid beta peptide, which leads to the formation of the amyloid oligomer. This cascade of events eventually degenerates the brain. And this work was published recently in the prestigious journal ACS Omega. An independent study in the transgenic Alzheimer's disease mice model by a team of researchers from the University of California published in the Journal of Alzheimer's Disease has supported the studies carried out by Dr. Mandal's group. Dr. Mandal and his team's upcoming human clinical trial is focused on enriching the antioxidant brain glutathione level that subsequently preserves cognition and arrests the progression of this dreaded disease. Dr. Mandal hopes their studies will help develop viable therapeutics to prevent the early emergence of Alzheimer's. Let's move to story number 2. As per the report of the United Nations, half of the fruits and vegetables produced are lost annually. Industries are trying to reduce post-harvest loss by using coatings. Wax coating is the most preferred way to preserve fruits and vegetables. Sometimes, wax is mixed with some chemical compounds that are known to be carcinogenic. Recently, many edible films have been developed using natural polymers to enhance the shelf life of foods. The edible films act as a barrier between the food and the external environment to prevent the direct interaction of food with atmospheric gases and microbes, which reduce the rate of respiration, keeping the food fresh for an extended period. But the cost of edible biofilms is high and restricted at the industrial level. The local fruits and vegetable vendors are not able to buy such costly biofilms. Researchers from the IIT Roorkee, led by Professor G. Gopinath, have developed a solution for dip coating and nanofiber coating using a blend of silk fibronin, PVA, honey and curcumin, a cost-effective method for fruits and vegetable vendors. The material used for coating is approved by US Food and Drug Administration. The technique utilized for synthesizing the biofilm are electrospinning and dip coating. The coating has been found to increase the shelf life of fruits and vegetables. The research is published in the journal Food Chemistry Molecular Sciences. Let's move to the next story. Researchers at the Indian Institute of Science, Education and Research in Pune have developed a interferometer-based atomic force microscope that revealed the viscosity and elasticity of a single molecule of a folded giant muscle protein called titin. The study could help understand and monitor small changes in protein structure and understand at the molecular level how some proteins manage multiple functions. Direct measurement of the viscoelasticity of a single protein molecule has not been possible so far. So scientists developed a special fiber interferometer-based atomic force microscope and used it to measure the viscous and elastic components of a titin molecule. They subjected it to an oscillatory load of about 10 billion times smaller than 1 gram. It produced an oscillatory deformation of 1 billionth of a centimeter, which is much smaller than the diameter of a hydrogen atom. The team, which included physicist Shiv Prasad Patel and Surya Pratap S. Diopa, applied force on a single molecule of the protein to measure its stiffness and internal friction. Beyond a threshold force, it moves from an elastic solid-like native state to a viscoelastic mechanical intermediate which dissipates energy. Such a change of state provides first direct evidence of internal friction and indicates how dynamic the protein is. According to the researchers, this technique could also be used to produce a data repository of other proteins by making a direct connection between their flexibility and activity. Now let's move to our segment Science Quest. The question for this week's Science Quest is, how long can you go without sleep? The longest time a human being has gone without sleep is 11 days and 25 minutes. The world record was set by American 17-year-old Randy Gardner in 1963. The effects of sleep deprivation are well documented, with symptoms including irritability, taking longer to make decisions, and cognitive rigidity, in which individuals can only think about things in one fixed way. Other side effects include loss of motivation, higher blood pressure, paranoia, memory issues, mood changes, visual problems, hallucinations, and difficulties with speech. Gardner experienced many of these side effects, including impairment of his cognitive and sensory abilities. He didn't consume any stimulant drugs to keep himself awake, but did have people around him to distract him from falling asleep. When the experiment ended, Gardner had been awake for 264 hours and 25 minutes. We have no verified examples of anyone staying awake longer than Gardner, and still we don't know for sure how long human beings can survive without sleep. With this, we come to the end of this edition of Science Time. Keep sending your feedback and suggestions through email. Our email ID is indiascience@vigyanprasad.gov.in. We will be back with more exciting stories from the world of science next week. Stay tuned to India Science.